Yo, hey, what's good, everybody? Hope that everybody is enjoying their weekend. Pretty sure you've been working hard on studying, getting things ready for this lecture test. I had hoped to have the test completely done, but I'm actually still working on the test. I went back and I thought I was done with the test. Went it back and I looked it over and I was like, nah, we need to edit this test a bit. So I want to talk about what I've done so far and what I know that I want to put on this test. And so, two things. There's two chapters for this test. You've got the muscular system, and then you've got the nervous system. And the muscular system was dealing with all that craziness going on inside of the cell, and then the nervous system, which was dealing with the different breakdown of the nervous system, the parts, the components, and then a lot of stuff surrounding the nerve cells. So let me back up and start with chapter 12, which is the nervous system, and we'll work our way through that. And then we'll talk about the muscular system and get into that a bit. So with the nervous system, we talked about the breakdown of it, like that there's a functional breakdown and then there's a structural breakdown. And with the structural breakdown, there was the central nervous system and then there was the peripheral nervous system. The central nervous system was the brain and the spinal cord, and then the peripheral nervous system was everything outside of that, which would be the nerves. You're talking cranial and spinal nerves. The functional breakdown is what I mentioned was a little more difficult. The functional breakdown was when you started talking about afferent versus efferent, where afferent is sensory information coming in to the central nervous system, and then efferent with an E is talking about motor information going out from the central nervous system. We said that there are sensory neurons relaying sensory information through an afferent pathway to the central nervous system, where things are computed, analyzed, you figure things out, you have thoughts, memories, feelings, assessment of incoming sensory information, and then after that assessment, you can then create a motor command or response which travels through motor neurons and on into through the efferent division. And we said that when things are going out through the efferent division, that they are either somatic motor or autonomic. And somatic is generally things that we can voluntarily control, like skeletal muscle, versus autonomic, which is automatic. It's stuff that happens involuntarily. It happens without thought. It happens without your guidance. It is involuntary activity, usually reflexive. And that stuff can be broken down into two groups. It can be broken down into parasympathetic or sympathetic. If it's parasympathetic, that's your way of automatically shutting something off or controlling the digestive system, controlling the body at rest. Then there's the sympathetic division, which is fight or flight. Remember we said parasympathetic was rest and repose, or rest and digest. And then we said that sympathetic was talking about fight or flight. It's increasing heart rate, increasing breathing rate, dilation of pupils, all that stuff to get you ready to either fight or run. Then we talked about action potentials in a neuron, and we spent a lot of time talking about the neuron. We talked about how a neuron creates an action potential. We said everything begins with a stimulation. So there has to be some type of stimulant, something that stimulates the cell to create an action potential. That can be electrical or it can be chemical. We said also where a neuron and another cell meet is called a synapse. And for the sake of argument, just about every synapse we ever talked about was chemical. So with a chemical synapse, there's a chemical or neurotransmitter that is released, it floats across the synaptic cleft from a presynaptic neuron and lands on the cell surface of a postsynaptic neuron. And then that postsynaptic neuron has chemically gated channels that respond to the chemical. Those gated channels open, they allow sodium to start coming into the cell. That makes areas a bit more positive. Now we go from resting potential to graded potential. That graded potential then stimulates voltage gated channels to open. And now the voltage gated channels allow lots of sodium to come into the cell, which makes things very positive, which therefore, if we see this influx of positive ions flowing in a direction, it creates an action potential, which that high millivoltage then stimulates other voltage gated channels, which then open, and they move in succession across a line. And because they're going in a line, they're going in an actual direction, and the direction happens to be on the axon of a neuron, that action potential is now becoming a current, and that current is flowing down the action potential all the way to the terminal end of the neuron, where at the terminal end, that is where we watched the animation where we saw the little yellow light get to the terminal end. It looked like a Hershey's Kiss, and it stimulated the end of the terminal end to have voltage gated channels that open that let calcium in. And then calcium came into the synaptic knob, that terminal end. We saw calcium come in. It made the synaptic vesicles move to the very edge, and the synaptic vesicles opened up and they released their chemicals, which for our conversation we talked about acetylcholine, ACH. And this acetylcholine was released from the synaptic vesicles out of the presynaptic cell into the synaptic cleft, and that allowed it to be able to bind to the postsynaptic cell. Some other things to clean up along there is we also saw where I got stuff popping up on my screen. Sorry. We also saw where we had to know like the stages of making an action potential. For example, when things were getting more positive, we said that the cell membrane was depolarizing. Then you made an action potential, and then we started seeing things shut down, and as the cell membrane began to get more negative, we said that it was repolarizing. Then because they opened so many gates and let so much potassium out, things got really negative, and we said it was hyper-repolarizing or hyperpolarizing. And then the hyperpolarization shuts those potassium channels off, and then the leak gates start letting sodium in, and then that brings it back up from -90 up to -70, where the cell membrane is at a resting membrane potential. That is essentially most of what we talked about for the nervous system. I have to go back and look at any little small minor details like when you're making an action potential, -70 is a resting potential. Then we slowly rise to about -55, -60. That's called threshold potential, and then that allows all those voltage gated channels to open, and then those channels open, and then that depolarizes all the way to +30. And once we get +30 millivolts, that's when we actually have an action potential. So then that also stimulates voltage gated channels to open and allow potassium to leave the cell, and potassium leaves the cell, and it starts making things very negative inside of the cell. And then once it becomes very negative inside of the cell, it gets all the way to -90, and then those voltage gated channels open, and not open, but they close, and they stop potassium from leaving, and they stop any sodium from leaving. And now we're back to the starting point again. Then there's the muscular system, and we talked about a lot of different things in the muscular system. Make sure there was on the board, I wrote 2 lists of terms, like our first time going into the muscular system. I wrote those 2 lists of terms. Make sure you know all those terms, because I will definitely pull questions that will be surrounding those terms. There are multiple choice questions. I'll have true and false questions dealing with the definitions of those terms. You need to know what a neuromuscular junction is and what happens in a neuromuscular junction. We had a PowerPoint that I sent you. Took me a while to get the PowerPoint to go through, but I finally got I sent it as a PDF, I believe, and it was labeled on how to build a neuromuscular junction. Make sure you know the components of a neuromuscular junction and the steps of things that happen in a neuromuscular junction. They're pretty much the same as what happens between a presynaptic neuron and a postsynaptic neuron. Make sure you know the general terminology for like the parts of a muscle cell, like a sarcolemma, sarcoplasmic reticulum, sarcoplasm, T-tubule, actin, myosin, troponin, tropomyosin, the role of calcium in a muscle contraction, the role of ATP in a muscle contraction. How do you make energy in a muscle cell? Remember, we talked about immediate energy, short-term energy, and long-term energy in a muscle cell. And how do you fatigue a cell? How do you create muscle fatigue within a cell? I think that pretty much wraps up everything that I'm going to be pulling to put on the test. So if you go over that, you should be good. If you haven't watched the animations or that crazy dude with the YouTube video, you're really missing out. You really need to watch that stuff, because if you just try to memorize as much of this stuff as you can and then close your eyes, say a prayer, and hang on, you could really not do so hot on this test. But if you can watch those animations and watch those videos and understand what's going on in those animations, what's going on in those videos, you'll be a lot better off, because these 2 chapters and what's coming on the test, they're really like stories. They're like movies. It's a process. When you get out of these 2 chapters, you kind of leave the process stuff and you go back to the magical land of memorization. So for those of you who like that, you'll be happy when we get over it. But in 211, there are several chapters that are like these, where you need to understand how things are happening. I'm not trying to be mean or anything to one of my classes, but one of my 211 classes in the past had a test in 211 on one of those chapters, and they didn't all do too hot on it, and I felt bad for them, because I think I should have told them that. I should have said, hey, if you're a type of person who usually memorize your way through a lot of this stuff and you do just fine, some of these chapters are not like that. Some of these chapters are really easier to understand if you understand the process. And the cool thing for you guys is that I already showed you the videos. The links are already on Blackboard of the animations and the videos that go through all this stuff. Now, what you want to be careful of, and I know this is a common student issue, I've dealt with it myself going back to school, is you feel rushed, you feel like you don't have enough time, so you just try to memorize everything, and then you wind up memorizing a bunch of garbage you don't need to know. Don't do that on this test. There is a lot of stuff in both of those chapters we never talked about. So you need to go back through your notes. If you're not a good note taker, if you took no notes, you need to contact somebody in class who took notes, because everything that I talked about was put on the board or posted. So if you don't have any notes, send the class, send an email out to the entire class. Say, hey, look, I'm looking for notes. Anybody got some notes that they can scan and send to me? Scan and text me, scan and load on an app or something. Hook a brother up. That's what you need to do, because you could be up all night long tonight trying to study everything and still not get through it all. But if you narrow the focus to just what we did in class, it's going to help you immensely. This is a lot of material, but it's just scratching the surface of what's all in those chapters. So I beg you, please stick to what we went over in class. If you have no idea what we went over in class, reach out to your classmates. Somebody copied the notes. I saw lots of people copy the notes. I saw lots of people write stuff down, take pictures and all that. I will say that's one of the dangers of just taking pictures and just sitting back and listening. If you don't have, some people can do that because the way their brains work, they just remember specific key things I say and they go back and they read it and that's great. But if you're not that person, if you really need to be taking notes, you really need to get those notes. So if you don't have those notes, please reach out to the class and try to connect with somebody to get what you can. Aside from that, that's all I got to say for the test. I'll see you guys tomorrow. Peace.